So yeah, I'm doing a little bit at the beginning and a bit at the end as well, but I have to say all credit goes to Rebecca. She's actually done all of the slides and all the prep, so just so you know. We can't possibly tell you everything that we do, so I'm just going to move that up a little bit, because we don't have time, but there's an awful lot of areas that we are doing research into, and this is why. Biosecurity is everywhere. We're never going to be able to be looking into everything that's out there, so we work very closely with KVH in terms of identifying where the areas we have to prioritise and where we do put our efforts. This is always being looked at. It's not static. And I'm going to just give you a real snapshot of the types of areas that we're working in. Oh, it's down there. That's better. So yeah, we're not actually going to talk too much about surveillance, Erin, but we are very interested in looking at a robust surveillance system, and that's working cross-sector. Wild kiwi fruit, absolutely applying some technology in there. Ceratocystis readiness, so working with the Brazilians on, because we can't work on it here, obviously. Neonectria as well, so very close partnership there with Plant and Food and Phytophthora. And then also, yeah, looking at some economics of this as well. I might get to touch on this later, but if I don't, I'd just say how important it is for us in Zespri Innovation to understand the value. And it's a bit like Ed was saying about the wine industry. It's an insurance policy, not a return on investment. So that's actually quite difficult to quantify. And so, yeah, that's why the economic analysis impact is really important for us to justify investment. I'm going to hand over to Rebecca's going to give you a bit of a deep dive into a couple of areas. Again, it's not all we do, but and then she'll hand back to me and I'll talk a little bit about how we're looking at innovation in the wider sense at Zespri.
Awesome. Thanks very much, Juliet. So, yep, like you said, I'm just going to go into a couple of different areas on a couple of the research projects that have been recently completed. So the first one is testing BMSB biocontrol research in China. So this was done by Plant and Food in collaboration with CABI. Yeah, so I'll just kick off. Oh, I went backwards. Okay, I'm going forwards. There we go. So the aims of this, so we're looking at biocontrol, so mainly the parasitic wasp, the Tricylicus japonicus. So the first objective was to assess the parasitoid abundance and diversity in kiwi fruit. We did this in 2 different orchards, organic and conventional, just to see whether those management practices had any impact. So the first objective is really looking to get that baseline within the orchard and see how much natural parasitism is happening within BMSB's native range. The objective 2 is building on that knowledge, so yeah, investigating how we can optimise that. What's the optimum release rate of this biocontrol to get the most control within orchards? Because this is one of the tools that we could release if there was a BMSB incursion to reduce the impacts to growers. Methods there, so like I said, 2 different Hayward orchards within China. The organic one was unsprayed, the conventional one was sprayed. That varied a little bit depending on the season, so anywhere between 3 to 6 sprays went on during that time. So I'll just speak to the objective 1 first. So we're looking at parasitoid diversity and abundance. So this was done in 2 different ways. One by gathering wild BMSB eggs. So we went out and surveyed those, collected them, grew them up in the lab, and then looked at how many BMSB hatched, how many parasitoids hatched, and what type of parasitoid they were. On the right side there, it's showing the sentinel egg masses. So this is where we grew up BMSB in the lab and then transferred them into the orchard. It was just another complementary way to gather data, as sometimes dependent on the levels of wild BMSB eggs, they can be hard to pick up. So yeah, that was the idea of the sentinel egg masses. Did the same as the wild ones 48 hours after we'd popped them into the orchard, took them out again and saw how many BMSB hatched, how many parasitoids hatched, and what type of parasitoids they were. So I'll just go through the diversity first, and then I'll talk you through the abundance. So we had slight differences in the wild BMSB egg masses. It was the majority Tricylicus japonicus, or the samurai wasp, which you might have heard of. This is the one that we can release in New Zealand, so it's great to see that this was the most prevalent in the wild eggs. Slightly different in the sentinel egg masses. There was Tricylicus japonicus and Tricylicus coltratus were the predominant species. Thoughts behind this is that there were more eggs out there, so there would have been less competition for those eggs. So Tristilicus cultratus is less efficient at parasitizing the BMSB eggs, so that's probably why that happened in this case. Ooh. Okay, talking through parasitoid abundance. So one of my first graphs. So on the right there, you're looking at the organic versus the conventional across the 3 seasons that we looked at, so 2018, 2019, and 2021. If the lines on the graph overlap, it means they're probably not statistically significant. So quite a bit of variation within those results, but I think you'll see that there is a slight trend for higher parasitism rates in the organic orchard compared to the conventional. Previous research has shown that if you apply chemicals to the parasitoid, this can impact their ability to lay the eggs in the BMSB. However, this was just done in 2 orchards, so the data set's a bit too small to make any conclusions. It could have been due to this, it could have been due to seasonal effects, it could have been due to site effects. So I think this gave us a really good understanding of what the natural rate of parasitism was in these Chinese orchards, and then in the next objective, sort of looking how we can optimize that. So we did a few different experiments initially to get an idea of that before we moved on to some field experiments. So in this case, we were looking at the ratio of wasp to BMSB egg mass. So we trialled a 1-to-1 ratio, so 1 wasp, 1 BMSB egg mass. Next one, 1 wasp, 2 BMSB egg masses, and the third one was 1 wasp to 3 BMSB egg masses. So you'll see there on your right, we tracked this in an organic orchard using exclusion netting that you can see in the bottom there. We did controls as well, so we had just the BMSB egg masses, so 1 egg mass, 2 egg mass, or 3 egg mass. And as you could expect, we had nearly 100% hatch rate for those, which you would expect if it wasn't being parasitised at all. You can see just on the right of the graph there, you've got the 1-to-1, the 1-to-2, and the 1-to-3. And you can quite clearly see that the 1-to-1 ratio is the most effective and really does reduce the hatch rate of those BMSB. So that's some excellent results. The other great finding is that even the 1-to-3 is really effective in reducing that to sort of around that 60% mark of BMSB released. All right, on to another part that we tested. We looked at the frequency as well of releases. So in this table here, you can see we've got different releases, day 1, day 7, day 14. So treatment 1, we looked at just releasing on day 1. Treatment 2, on day 1 and day 7, and then treatment 3 on day 1 and day 14, and then treatment 4 releasing on day 1, day 7, and day 14. So you can see from the graph here, and we did test this in May and August, this just aligned with when the BMSB life cycle started, so when the most eggs were available to parasitise. So you've got mean observed parasitism up the left-hand side there, and you can quite clearly see treatment 4 was the most effective, so releasing those wasps each week for 3 weeks was the most effective. So I guess what you're probably all wondering is what does this mean for us? How much does this actually reduce the impact that BMSB is going to have on the orchard? So that's what we did. We used the data and some more data that I haven't been able to go into here just because of time constraints to see how much damage was done in a kiwifruit orchard in a release and in a control orchard. So we did this in 6 orchards, all organic, all Hayward, 3 controls where we didn't do any extra releases, and then 3 where we did do the releases. So you can see in the bottom left here, the far left graph you're looking at is feeding damage. The one on the right is the number of fallen fruit, and the control orchards are the circles, the release orchards are the triangles. And you can quite clearly see that the triangle, the release orchard, is really reducing the impact of BMSB to fruit damage and of BMSB to fruit drop. And averagely, so the control orchards were 40% damage to fruit, the release orchards were 20% impact to fruit. So that really shows how optimising the release of this parasitoid is a really good control mechanism. And in my conclusions, yeah, this is going to be really useful if we get a BMSB incursion to help reduce the impact to growers and really manage that pest in a very sustainable manner. And this wasp is incredibly specific to BMSB, so off-target impact should also be nicely limited there. One of the things that we did know is although we modelled the perfect scenario of releases, we were actually slightly limited by our ability to produce the amount of wasp required. So I think we all know that will be a limiting factor no matter what, and dependent on the size of incursion and the length of time, getting our hands on that wasp is going to be one of the limiting factors. And I know the KVH and the GIA BMSB Council are working on how to acquire and rear and make sure we have really good access to that wasp if we need it. Okay, so spotted lanternfly. This is the second piece of research I was going to talk to today. So we recently completed some work in China. This again was completed by Plant and Food Research in collaboration with CABI. If you weren't here last year, you might not know what it looks like. So I thought I'd just show it here. It's just there, just underneath BMSB on the KVH most unwanted list. This is because it's a highly voracious polyphagous phloem feeder. So yeah, it can cause some significant damages. It's beautiful up on the right-hand side there, but the lower picture, I don't know if you can see clearly, but that's a load of spotted lanternfly there swarming on that apple orchard. So I'm sure we wouldn't like to see anything like that in our orchards. I'll probably refer to spotted lanternfly as SLF from now on. Sorry for the acronym, but it gets quite long after saying it for a while. So the aims here was to understand the phenology of SLF in kiwi fruit orchards, and to also understand the impact of spotted lanternfly on kiwi fruit orchards. The methods, so we looked at 2 kiwi fruit blocks, one Hayward green and one gold orchard, and we were using visual inspections for this, so using a W-shaped pattern through the orchard. So these are the different life stages of SLF. You've got 4 nymphal stages, an adult stage, and then an egg stage. So this is what it looks like on an orchard, which is pretty cool. So you can see the early spring, sort of the nymphs starting to hatch out of those eggs. As you start to get that bud development, you've got your further, second, third instars, and then on the bottom left, you can see your 4th instar nymph and your adult there when your kiwi fruit's nice and mature. So numbers on the orchard, we didn't see a whole heap of eggs. They're so small you can't see them on the graph, but the Hayward orchard had 5 eggs, the Gold orchard had 12 eggs, and it was there was 44 eggs sit in the neighbouring block. So they were there, but in the blocks we looked at, there weren't that many eggs, and a lot more nymphs there. But I'll show you this in the next graph because this shows it a lot better. So this is a heat map. The darker the colour, the higher the number. So you can see here with the eggs, you've got them, once the eggs finish, you start to see the nymphs, and then around that 4th instar nymph, you get a sort of decrease in numbers. Now this is because at this stage they get very mobile and they will start to move in the orchard, they become harder to survey, and they may be moving on to different crops to feed on as well. And you can see here this gap at the top where the adults aren't present in either the green or the gold block. We did look in this study to see if there were any other host crops, and they were present on host crops the whole year round, and because of that highly mobile nature, they could have been coming in and out the orchard, but just not seen by the surveyors. So that's the theory, is that the SLF in the adult phase, that's when they're most voracious feeding, and they'll probably move on to their most preferred hosts like the tree of heaven if it is available. So impacts, so these are phloem feeders, so they can cause impacts as nymphs and adults by feeding on the canes, and then they can cause impacts as adults from the honeydew that they excrete, which then can turn into sooty mould, a little bit like your passion vine hopper does now. Apart from this insect's quite a bit bigger, so it's likely this would produce quite a bit more honeydew and therefore quite a bit more sooty mould. Strangely, this is not what we saw, because we didn't really have the adults in the orchards. We didn't see any honeydew, we didn't see any sooty mould. This doesn't mean this won't happen in New Zealand though, depending on what different hosts are available for the spotted lanternfly to feed on, and just dependent on the environmental factors of sooty mould development. Then looking at the canes, we couldn't determine what damage was caused by spotted lanternfly and what damage was caused by other insects like BMSB. Just because this was an open field environment, it was very hard to determine. So, is it good? Yep, so next steps. So because this told us something about the phenology and when it's present in orchards, but unfortunately because it was the open field trial and the pest pressure was quite low, just the 5 and the 12 eggs found, we'd need to do some lab studies or using some of that exclusion netting type experiment to see what the real impacts are, especially at that low, medium and high pest pressure, and then assess feeding damage. And we're looking to do some testing of our current crop protection chemicals to see whether any of those would work, just to sort of limit that impact in the nymphal stage, because realistically it's a new environment that spotted lanternfly would be coming into, and it could cause some significant impacts, at least in the initial stages of an incursion. And with that, I'd like to hand over to Juliet to sort of go into what we're doing next and how our portfolio is looking forward.
So yeah, I've been at Zespri for nearly 10 years, and that back then when Dave Tanner employed me, thanks Dave, it was a much smaller effort in innovation, but with that growth has come more people, more resources, and more funding, but we still have the same constraints or the same, I suppose, discipline around how we spend that money. So we have to make choices always. This year we probably had to make a few more choices than others. But we have actually had a really big overhaul of our innovation process just recently, and we refer to that as our operating model. And all of our research is outsourced. So for those of you that don't know how innovation works, at Zespri we have innovation leaders like Rebecca, and then we have research partners like Partner Food, for example, and we have strategic partners like KVH that we work with in some areas like biosecurity. And Zespri ring-fences innovation spending as a percentage of revenue. So that's good. As Zespri grows, in theory, there's more money available. And within that innovation budget, we actually ring-fence for resilience. And biosecurity is one of the portfolios that sits in the resilience pillar, along with climate change. So that means that there is a certain amount of guarantee that we will keep investing in biosecurity. And that's like quite apart from, I suppose, the grower sentiment is always very, very strong for investment in biosecurity. It's increasing in climate change as well, I think. So anyway, our new innovation operating model is it has been reviewed, and I suppose it's a bit of a theme that's come across in the last couple of days around organisation, around having structure and governance around what we do, and that's in order for us to make the best use of our resources, people and money. So we've got this new structure in place. Some parts of it are about engaging more with the business. Any of you that have worked in any sort of science area in a business will understand that sometimes they're seen as a little bit apart from the rest of the business, but what we've tried to do is to get greater engagement and ownership up front from the business units that use our research. We're kind of a non-critical function in times of like most of the year. We're not BAU. We're sort of apart from that, but that doesn't mean that we don't need to engage. We do need to engage. We need to be doing things that are relevant, because actually the impact of the work we do is out of our control. It's taken up by other people. So it's taken up by the extension team or the technical team or the marketing team or KVH or other people. So yeah, part of the philosophy around changing this model was around getting better engagement, making sure what we do is relevant. And one of the main activities that we do is called a sprint process, which is this first box here. And what this is really about is a strategy review or strategy refresh. Some portfolios like climate change, we haven't got an existing strategy, but for biosecurity, we have a long history of having a strategy and having a review of that strategy. But in the new process, we're putting a bit more, like instead of having a day of strategy refresh a year, we've had the last 3 months of a strategy review and refresh, and it's called a sprint. And I'd say, again, I just want to call out the very strong partnership with KVH in that process. We took a lot of Matt's and Erin's and Leanne's time, as well as many, many other people in the room. Sorry, I'm not going to mention you all, but we really appreciate that. And the purpose of that is really to set up for the next 2 to 3 years. So what are our key challenges? What are the key challenges that the industry is facing? Then what are the key areas where we can form programs to help with those challenges? And then, okay, what are these work streams? Who might we be able to work with to deliver those, because it's all outsourced? So we've just completed that process, and it's been really, really successful, I'd say. So yeah, we've had a lot of alignment. We've had a lot of input from different people. We've got our programmes of work as a draft form. They've not yet been signed off by our exec and board. That's the next step. And then, so that's the first stage. And if you imagine, if we all started from a standing start, we'd do that first and then we'd set off. We're already running the race though. And so we have also got lots of programs of work in progress already. So these different stages you can see of the process are already populated. And we're not going to stop doing things that we've got in flight, but it means that as we churn through, eventually we'll be doing things from scratch, from ideation through development, validation, launch and evaluation. I think that was all I was going to say on that one. I won't go into detail of that because that's just the detail. But yeah, I suppose where we've landed is we've got a series of programs. You can kind of split it into mitigation and readiness, maybe. But yeah, I suppose there's some of the new things that are coming up would be around emerging, new technologies. I think this is something perhaps we haven't put as much into in the past that we will in the future, because I think this is an area across the whole of innovation where we're seeing lots is happening in the world. We need to bring it into biosecurity and really capitalise on that. But yeah, these, as I say, these programs will be confirmed in the next couple of months when they get signed off, and we will continue to work in this area. So I think that was all I was going to say. We've probably got a few minutes for questions, have we? But no? Okay. But thank you. That's it.
Thank you,
Thanks.